Litochrome lacustris specifica is a common euryhaline brackish water copepod in order Herbaticoid. This Herbaticoid copepod has been used in the studies worldwide because of the high reproduction rate, short life cycle that can tolerate wide range of environmental parameters. The objective of this experiment is to observe the egg production rate and hatching success of the nobly, and to determine the growth and survival rate of this copepod from eggs to adult. Low pH level in the ocean has adverse effect towards the copepod. Ocean acidification seems to give physiological effect in their reproductive stage, thus affecting their growth. So, for this experiment, we propose the effect of the pH towards the growth of this Herbaticoid copepod. The significance of this study is to measure what pH is considered harmful towards the growth of the copepod. We choose pH 4 for the treatment and pH 7 for the control. However, in the middle of the experiment, we change pH 4 to pH 6 because the microalgae, which is the food for the copepod, die because pH 4 is too acidic.
I am Rin. Now I would like to talk about the methodology for this experiment. Before that, we need to know about the material and the apparatus. First of all, we need 100 ml beaker, 600 ml beaker, pipette, seawater, vinegar, pipette filler, pH meter, distilled water, hemacytometer, petri dish, dropper, dissecting microscope, compound microscope, syringe, conical flask, measuring cylinder, micropipette, aeration rod and tube, aluminium foil, parafilm tape, hand tally counter, and last but not least, cell culture plate. Methodology. First of all, the copepods were given with the same amount for each group and been separated into two types, control and treatment solution. However, the solution for the copepod has been changed to pH 6 with 0.2 ml of vinegar and 0.18 ml of nanochloropsis from pH 7. Next, step 2. The control and treatment solution were placed into the aeration chambers for 2 days. Step number 3. 15 copepods in both solution were sorted into the two culture plates with 10 ml of each solution using dropper. Next, step number 3 need to be repeated with the copepods from the treatment solution. Step number 5, the egg production rate of the copepod was observed. Next, step number 6, the eggs that were successfully hatched need to be calculated. Step number 7, the data collected was recorded into the datasheet. Meanwhile, every picture for the data collected was taken. However, if the egg sac detached from adult copepod, it was removed immediately from the well. Step number 5 till step number 7 need to be repeated continuously for every day until the experiment was done. Lastly, rate for the eggs analysis. The data need to be analyzed by using Microsoft Office. Egg production rate, EPR, equal to the CS times S times 24 over time, where CS equal to the mean clutch size, S is the ratio of number of females releasing eggs to the total number of females in the population. E is the duration of incubation, about 48 hours. Meanwhile, for the hatching success, the formula need to be used are number of eggs hatched over number of eggs produced times 100%.
We move on to results and discussion. I am going to talk about the egg production rate of Nitocra lacustris pacifica. The mean egg production rate of Nitocra in pH 7 is higher compared to pH 6. The mean number for pH 7 is 213 eggs, while pH 6 is 108 eggs. Hapag-icoid copepod is a brackish water copepod where they are adaptable to variety condition. They can live in fresh water with the range of pH 6 to 8. They are more tolerant to higher pH and they can produce more eggs. The previous study shows the egg production rate is lower in low pH. Hence, it shows that the copepod prefer pH 7, which they can produce egg more than pH 6. The treatment pH 6 is considered in acidic level, which the copepod may be in the stressful condition, hence it lower the egg production rate.
We will move to the hatching success of nucleus and the number of individual nucleus survive. The higher percentage hatching success of nucleus is pH 6, which is 70.37%, and percentage for pH 7 is 41.31%. This formula are used to calculate the percentage of hatching success. For the individual nucleus survive, pH 7 has the higher number with 65 individuals on 4th day and the lower number on 5th day with 5 individuals. From the previous study, it stated that at high pH, nucleus production rate was increased. Same goes to nucleus production at low pH, where the production of nucleus is decreased and had adverse impact. At first, we used pH 4. Unfortunately, Nitocra lacustris pacifica cannot survive and died, so we decided to increase the pH to pH 6. To compare with previous study, it shows that the copepod performed better at high pH. From the graph shown, we can see that hatching success is more higher in pH 6. This is because some eggs were not hatched. The nucleus survive is higher in pH 6 on first day, but decrease and continues in the following days, while number of nucleus survive of pH 7 is increasing. Thus, we can tell that pH 7 can significantly delay eggs hatching success instead of reduction in eggs.